Good morning, everybody. I'm Yvonne. I'm a scientific database curator at UniProt, and I will guide you through the webinar today. I will present you one of our current curation projects on proteins involved in neurodegenerative disorders, and I will show you how we try to increase our understanding of those diseases through data curation. At UniProt, we provide the scientific community a comprehensive, high quality and freely accessible resource of protein sequences and functional information. We aim to provide expert literature curation on detailed information on protein function, interactions, pathways, and more. We provide primary protein sequences, including isoforms, disease variants, and post-translational modifications. Importantly, we provide stable identifiers to identify specific proteins and which should be used when citing UniProt entries. Neurodegenerative disorders are a heterogeneous group of diseases characterized by progressive degeneration of the central or peripheral nervous system. They cause problems in movement, so-called ataxias, or in mental functioning, so-called dementias. The dementias are the biggest burden of neurodegenerative diseases, with Alzheimer's disease representing approximately 60 to 70% of all dementia cases. A combination of genes and environmental factors contribute to the risk of a person to develop a neurodegenerative disease. That is, a person might carry a protein variant that makes them more susceptible for a neurodegenerative disease. Recent advances in genome sequencing are contributing to our understanding of underlying causes of these diseases. However, it still remains a challenge to establish the relationship between genomic variants and the disease phenotype. Our aim is to create a link between genomic variants and disease phenotype and thereby contribute to the understanding of disease development. For the current curation project, our curators have identified relevant neurodegenerative diseases by literature search and search on disease-dedicated databases such as OMIM and Orphanet. We then annotate disease-associated variants found in patients and assess their pathogenicity based on guidelines by the American College of Medical Genetics and Genomics. We review the relevant literature for those proteins involved in neurodegenerative diseases for the human proteins, as well as the orthologs in model organisms such as mouse, rat, and Drosophila melanogaster. We then collect molecular data for these proteins on protein function, molecular interactions, localizations, post-translational modifications, and protein structure. For the current project, we have currently identified over 200 neurodegenerative diseases and 165 associated proteins. And here I show you a short list of the identified targets and highlighted in red 3 examples of proteins that I will shortly talk about to exemplify our curation efforts on the impact of disease variants on protein function and structure. To access protein entry, you can go on the UniProt main page and search in the free text search bar, or use the advanced button to the right for a more specific search using search terms such as Alzheimer's disease, for example. You can also use the Access tabs below, where you can access different UniProt resources. You can find diseases annotated in UniProt in the Disease Access tab, where the diseases are listed in an alphabetic order. Looking at the main section of the UniProt entry for human E3 sumo protein ligase EGR2, we find following information. We find the protein accession number, the protein name, the gene name, the organism, as well as different sections with information, for example, molecular function and involvement in pathways in the function comment. The axis tabs to the left allow you to jump to different sections of the entry and to choose the content visibility. The function comment is a free text format written by our curators after careful assessment of the relevant literature, and the sentences are tagged with evidence tags. We use experimental evidence tags if the experimental data is from a publication using their PubMed identifier, or similarity tags if the information is obtained from a protein ortholog in another species using their UniProt identifier. A list of all publications linked to this protein entry can be found in the publications viewer on the left side. Here, all publications linked to the protein are listed, and the reviewed publications are tagged with a gold standard tag. These are manually curated publications by the curators. Further, you will also find computationally mapped publications which provide more information about the protein, which has not been reviewed by our curators. In the publications list, you will find information that has been obtained from the reviewed publications, such as disease information or variant information, as well as hyperlinks to the articles in PubMed and EuroPMC, as well as other UniProt entries which cite the same article. To the left, you find categories for the reviewed articles, where you will find specific information you might be interested in, such as pathology information. Importantly, the users have the possibility to submit their own publications of interest. And this option you find on the UniProt main page under UniProt data. Click on Submit Your Own Data and choose Associate the publication and annotation to a UniProt entry. With that, you can contribute the references of interest to the proteins and help us find relevant papers to curate. Back to the main view of the EGR2 protein, you find information on disease involvement under pathology and biotech. Here, the diseases are listed, and you find disease acronyms and the disease name, as well as a disease description of the disease the protein is involved in. The disease description is from the literature and in a controlled vocabulary if the disease is also described in OMIM. Before the disease description, there's a note that shows the role that the gene protein is playing in the disease and distinguishes, when possible, between susceptibility, disease-causing, or modifier gene. These following variants are annotated in UniProt. We annotate functional variants, that is mutations in the coding region of the gene, including missense amino acid substitutions, nonsense substitutions producing a stop codon, small in-frame insertions, and deletions. The variant pathogenicity is then interpreted based on guidelines by the American College of Medical Genetics and Genomics, and distinguishes between three different categories: disease-causing variants, variants of unknown pathological significance, and neutral variants, so-called polymorphisms. The manually curated variants can also be found in the pathology and biotech section, and they are listed according to their disease association. Following this variant information can be found here: a UniProt specific variant identifier, the variant position, the amino acid change, the disease association, the variant-specific comments, such as impact on protein function, variant evidences, and database cross-references to dbSNP and ClinVar, for example. If you're interested in the impact of a variant on protein structure and function, you can go to the feature viewer, which is found in the display to the left, where protein features are displayed in a visual manner. The features are categorized in different sections, such as domains, sites, and variants, and by clicking on the category header, the section is expanded below. So you can click on variants, and it will show you all the variants associated with this protein. The variants are color-coded according to their involvement in disease. The red variants are pathogenic variants, the light green are likely benign variants, and the dark green are of uncertain status. The blue variants are imported variants from large-scale studies such as COSMIC, EXAC, or 1000 Genome Project. You can also filter for the variants of interest. For example, you can click the filter on the left to choose for only the pathogenic variants to be displayed, which will be shown in red. By clicking on a variant, a box will show further information such as the position of the variant, disease association, and the publications with the PubMed identifier. Further, it will highlight in yellow a possible overlap with other features of the protein. By clicking on the features above, you will find all the variants that are lying within this feature sequence highlighted in yellow and highlight a potential hotspot of pathogenic variants, like here, a hotspot of pathogenic variants lying in the zinc finger domains of the EGR2 protein. Using another example of presenilin 1, a key player in Alzheimer's disease, I would like to show you how to visualize the impact of a variant on protein structure. The variant of interest is variant 435 with a leucine to phenylalanine change. And in the feature viewer, you are able to see that it lies within the PAL motif of presenilin 1, which, according to literature, is required for the enzymatic activity of presenilin 1. If you're interested then how the variant might impact protein structure, you are able to see published 3D structures of proteins in the feature viewer. Published 3D structures are imported from PDB, and you can select your structure of interest in the table to the left, and the protein structure is displayed, where the variant amino acid is highlighted in red, thereby showing you a potential impact on protein structure formation. And this information might be of interest for researchers involved in drug development. On another note, within the current Curation project, we are specifically interested in rare neurodegenerative diseases. Those disorders affect less than 5 in 10,000 individuals in the general population. And by compiling available information on proteins associated with rare disease, we hope to help researchers to a better understanding of the relationship between protein function and disease. And for that, we have a special focus on orthologs in model organisms to gain more insight into protein function. And here, I just would like to show you how we apply ortholog identification in UniProt with the example of SERF1. So human SERF1 is involved in a rare neurodegenerative disease, And you will find information on this protein, such as function, disease involvement, and disease variants in a UniProt entry. We then apply bioinformatics and functional analysis to identify orthologous proteins in other species. And we group them into protein families and try to name them with the same protein name for easier identification. The protein orthologs are then annotated as well based on relevant literature, and we add following information such as function, gene ontology terms, as well as disruption phenotypes. In the disruption phenotype, we annotate knockout and knockdown information, as well as mutagenesis assays with more specific amino acid changes, which might give valuable insight into protein deficiencies and disease development. Here, a short list of rare neurodegenerative diseases where we could identify one-to-one orthologs in the fruit fly Drosophila melanogaster, and where our curators are currently working on. Finally, for those who have an interest in a more large-scale analysis of variant data, programmatic access of variant data is available. You can find that on the main UniProt page under UniProt data, programmatic access, where you can access variant data such as genomic locations of reviewed variants and imported variants from large-scale studies under the Proteins REST API. And with that, I would like to end this webinar, and I hope that I could give you some insights into our current curation project and how we try to find new insights into the molecular function of proteins associated with neurodegenerative diseases and disease mechanisms, how we try to gain insights from orthologs in model organisms, and link molecular defects to disease phenotype. With that, we hopefully are able to provide an overview for researchers in the field of neurodegeneration, clinicians, and biomedical researchers. I would like to thank the whole UniProt team, which is spread across 3 different sites at EMBL-EBI in the United Kingdom, at PIR in the US, and at ZIP in Switzerland. I would like to acknowledge our funding bodies and thank you for your attention.